Good afternoon, everybody. I'm Jerry Vockley. I'm the Chief of Genetic and Genomic Medicine here at Children's Hospital and Director of the Center for Rare Disease Therapy. Thanks for joining me this afternoon for our regularly scheduled, although irregularly timed, webinar series. Today, I am going to present to you an overview of gene therapy and actually more broadly nucleic acid therapy for rare disorders or genetic disorders that I hope you'll find interesting. I'll try to keep the geek science down to a reasonable level because I'd also like to talk to you a bit about the challenges that our system and any healthcare system has in moving forward to deliver gene therapy, as much as I will, as I want to talk to you about some of the concepts of gene therapy. So hang with me. If the science is a little dense for you, there'll be some stuff coming back up at the end that you may find a little bit more palatable. So. Working in a different system that we haven't used before, so we got to make sure everything works correctly. When we talk about genetic therapy or therapy for genetic diseases, there are a number of ways that we can attack trying to reverse the problems related to a gene defect. We can try to augment a missing function, and that is what a lot of people typically think of when they think of gene therapy. If a gene is broken, give them a new copy of it, or give them new cells. That's a liver transplant, right? You can also, though, replace the product of that gene. In this here would be a protein replacement or substitution. And many of our lysosomal storage disorders have protein replacement therapy. PKU, another disease that we deal a lot with in the center, has enzyme substitution therapy in the form of pegvaliase. And then we can also try to make variant proteins that that don't work properly and don't fold properly, a part of natural protein development, try to fold better. And we use compounds that are called chaperonins for that. These are just molecules that help proteins fold. We can try to reduce the reliance on that pathway. That is, we can either manipulate the environment, especially in the context of diet, to not expose the individual to the things that they can't use, or we can try to redirect whatever it is that's the toxic compound away from that pathway using the various drugs. And then finally, we can try to eliminate anything that's toxic that accumulates. This is something that we do, for example, with fatty acid oxidation disorders and carnitine, or rather organic acidemias with a drug called carnitine, or with urea cycle defects with drugs that bind up the ammonia that accumulates in those disorders and causes toxicity. And so today we're going to spend a little bit of time looking at the process of developing modern therapies for rare diseases, and then talk a little bit about those therapies, and then come back to implementation of therapies. So part of the problem with developing treatments for rare diseases historically is that they were really it was very, very difficult to identify drugs that would attack those diseases. That's because traditional drug screening is undirected. You start with 100,000 compounds or 1,000,000 compounds, and you just sort of test to see if they work. It has absolutely nothing to do with the mechanism of action of the disease or what we know about the disease, and that's a very expensive process. Instead, as as we move towards development of therapies for genetic disorders, what we are doing is trying to use our understanding of the processes that are causing that disease and target the therapies to reverse that. And this allows us to sort of shortcut all of that screening that you need for in the early going in a random screen or an undirected screen, and go right for where the problem is. So it's a new way of developing our therapies, and it also requires that we have a novel approval pathways with regulatory agencies like the FDA. This is a classic view of drug development. So as I mentioned, you take a whole bunch of things and you test them in this first stage called drug discovery, and then you find some things that sort of work okay, and then you do some additional manipulations, and you find something that works a little bit better, and maybe you try some early clinical trials, but mostly you do cellular and animal work until you get the compound that you're going to move forward for clinical trials and regulatory approval. And this is a very inefficient and expensive process, and it takes a lot of time, typically 10 to 15 years. And that's a long time when you're sitting there thinking, we've got 7,000 genetic diseases that we want to develop drugs for. So we need a better approach. The way we're looking at this now in genetic disease is to go back to our very basics. For those of you who have had biology, I think they teach this in junior high school now. It was in college when I was at that age. Something called the central dogma, and the central dogma says that our genetic information is carried in our DNA. That DNA is used to make a copy of itself into a molecule called RNA, and that RNA then actually carries the cell uses that information to make our proteins. Now, it is a little bit more complicated than that, and over the years, the process has been filled out to cover a number of other steps. First of all, DNA is not all the same. There's DNA that is basically just the 4 letters of the DNA alphabet hooked together, but there also are places in the genome, your genetic information, where the DNA is modified, and so that changes how it can be expressed. Regardless, what has to happen is that every time a cell divides, the DNA has to be copied. That's called replication, and there are vagaries about that that can be affected in genetic disease. Then it has to be transcribed. That's the process of making an RNA copy. Now, it turns out that almost all genes in our body are not a single string of information. The RNA that is made has coding sequences that have information for protein in areas called exons, but then there's a lot of information in between it in the introns that has to get eliminated before it can be now in a mature form and used to make protein. That process is called splicing, and there are lots of genetic diseases where impairment of that splicing process can lead to the disease. But if a mature RNA is made, now you have the messenger RNA. That's called that's the mature message, and the body can read it. The cell can read it 3 nucleotides at a time to add an amino acid. Now that to make a protein, those amino acids are added one at a time, and so it's sort of like stringing beads on to make a necklace. But the problem is that proteins aren't two-dimensional. That is just information lined up on a string. It's three-dimensional. You have to fold that string of beads into the right conformation to make the protein active. And so there are mutations where you can affect the function of the protein, but there are also mutations where the protein can't be folded properly. And you can have therapies directed at all of these processes. Once you have a candidate drug, now you have to prove that it works, and this is the process of clinical trials. In the context of common disorders, usually that's done by taking a group of patients and splitting them into two groups and giving half of them a placebo, an inactive compound, and half the test drug, or half a standard of care medication and the other half a new drug to test. And you compare the two. We can't always do that in rare diseases because sometimes it's just not ethical. If we know a patient is going to die from their disease in the first year of life, you're not going to give that child a placebo. You're going to want to give them the potentially functional molecule that will affect that disease. And so you have to have a different paradigm for doing these kinds of clinical trials. You have to know what the natural history of the disease is, and then see if your treatment changes that natural history. So if you know that patients have, in the worst case scenario, they die in a year, or in milder diseases, they develop certain symptoms at certain times or at certain frequencies. Now you give everybody the drug potentially, and you see how that affects them. And so these are alternate designs that have to be discussed with the FDA in advance before you can even move forward, because otherwise you might get to the FDA at the end and they'll say, well, we don't like that design. You got to go back and do it again. And then thinking about what the endpoints of your trial is important. Ideally, what we look for is what I just mentioned. That's the alteration of the disease course. But sometimes diseases have a very long course. What if you don't see it takes 20 years to see the changes, but you need to start treating at the beginning before you can have an effect at the end? Well, you can't wait 20 years to test a drug. So we look for markers that try to tell us that something's happened, and we call those biomarkers. Something that changes in a cell in your body that we can measure that now is altered by treatment with the drug. And because it's hard to know how well that's working clinically over time, what we then have to do is if we can convince the regulatory agency that drug looks like it's working, then you can get approval, but you then have to do longer term studies after approval to continue to monitor the clinical efficacy of the drug. So now that you have a little bit of an idea of the process that gets you to a therapy, let's start talking about nucleic acid therapies. And there are a whole variety of different types of nucleic acid therapies. We're going to spend most of this time talking about gene therapy, and in fact, we're really going to be talking about gene addition. That is supplying a functional copy of the gene to replace that copy that's not working in a patient's cell. You can also provide mRNA. You're all familiar with Moderna. They make mRNAs, and right now, you know, their big focus is on providing mRNA vaccines. But before the COVID hit, we and others we're working with them on mRNAs that would treat rare diseases. Now, mRNAs don't hang around very long, and so unlike DNA, which can stay around for years or even decades, the mRNA needs to be replaced every couple of weeks, and so that's not a permanent solution. if you can give a DNA copy of a gene, that gene can integrate into the cellular DNA, and then that way the information is passed along to daughter cells over time as they divide, and that's a permanent fix. A lot of the therapies that are being under development now use a different approach that instead of integrating, the DNA hangs around in the cell, but just sort of sitting there by itself in something called an episome. And these would be the AAV therapies, the AAV vectors that you'll hear so much about in this talk and in the public, the lay press about gene therapy. And the problem with that, with episome based therapies, is that over time there's the possibility that they can be lost. Certainly every time a cell divides, only one cell gets a copy of it because you only have one copy of it. So it just randomly goes to one cell or the other, and over time you tend to lose it. There's been a lot of press about CRISPR technology, gene editing technology. That actually goes in, and instead of just giving an extra copy of the gene that now works, you try to actually correct the copy of the gene that's within the cell. And there are clinical trials now for genes that do just that. We're not going to spend much or really any time talking about some other alternatives. The mRNA therapy that I've already mentioned, something called allele-specific oligonucleotides. These are short pieces of DNA that can target the cell's own DNA and sort of upregulate it or downregulate it, and make more or less of a gene depending on the disease and what the mechanism of action is in the cell for the problems to develop. Or you can affect that splicing process that I talked about earlier, and if the splicing is not occurring properly, then you maybe can fix it, or you can modify it in a way that makes at least excuse me, a partially active protein. So as I said, we're gonna talk more here about gene therapy. The goals of gene therapy are to achieve a durable expression of that therapeutic gene that we're delivering at a level that's sufficient to hopefully cure the disease, or at least make it better, make it good enough that it's worth the therapy. The therapeutic requirements of that mean that we have to get the gene into the relevant target tissue. So if it's a disease of the brain, you got to get the vector into the brain. If it's something that affects the liver, you want it in the liver. If it's necessary in all the tissues in the body, then you have to have something that's a therapy that's targeted broad-based. We've already talked a little bit about the stability of expression, but another very important aspect of gene therapy is that it can't interfere with the functional integrity of the cell. You can't cause another problem by fixing the first problem. Now, ideally, it's meant to be curative and permanent, but at least some of the vectors that are out there have the potential to be lost with time, though we're talking about years, likely 5 or 10 years, as opposed to short term. So even if you can get 5 or 10 years of improvement, that's probably a good enough outcome to move it forward. There are a number of products already approved, greater than 20, that most of these are related to cancer therapy. And there are thousands of clinical drugs under development. If you go to clinicaltrials.gov, the best place to look for things like that, you can actually come up with greater than 3,000. Most of those are in the preclinical phase, and I'll talk about that later. And relatively few are actually going into patients. Most of those are cancer related, but we do have, probably a dozen at least, maybe even more than that now, of gene therapies that are being tested for rare diseases, for genetic disorders. To understand where we're going, it's important to understand where we've been. And so I'm going to talk a little bit about the history of gene therapy. and why we do what we do, and where we are in the process now compared to when it first started. So, you know, DNA was first recognized as the molecule that carries genetic information back in the 40s. It wasn't all that long ago from an evolutionary standpoint, I guess. In the late 40s, the early 50s, even going into the 60s, a lot of the studies were focused on trying to prove that certain pieces of that DNA were active or not, and oftentimes that meant transferring it from one organism to another. Something called transformation, and usually that was done by a virus, and that was the beginning of what we now see as gene therapy. There's a disorder of immune function called adenosine deaminase deficiency, and that received early attention because it was felt that if you could just get the gene in the blood cells, which are easy to get to because they're in the blood, it might you might be able to cure that disease. And there were a lot of early studies looking at a variety of different viruses that were engineered to carry that gene, that were used in the early years of gene therapy. However, for a variety of practical reasons, all of those have been replaced by other vectors that don't have as many adverse events or side effects. Because we knew the cause of the genetic cause of the many rare diseases, even back then, those rare diseases, and especially biochemical disorders, inborn errors of metabolism, were the most frequent early target for gene therapy. However, it had a slow start. Here's a little bit of a timeline, and up above the timeline, there's some important hallmarks of development of the sort of regulation or recommendations for genetic therapy. So back in the 60s, we started newborn screening. In 2011, the recommendation for broad-based carrier testing for cystic fibrosis was made and incorporated into many prenatal programs. In 2013, the American College of Medical Genetics put out the first version of what we now recognize as the modern-day newborn screening panel, and then just recently there's been a call for population-wide screening for chromosomal problems in babies by testing the blood of mom. Now, and a major point, a turning point in all of that was the completion of the Human Genome Project in 2003. Now let's look at the development of therapies. Remember, I talked about enzyme replacement therapy. Well, the first one of those was only approved back in the early 90s. And most of the rest of the last decade of the 2000s and the first decade and a half of the 21st century were focused on an enzyme replacement therapy. In 2016, we had the first FDA drug, nucleic acid-based drug approved. This was mRNA therapy. It's actually not replacement therapy, but it redirects splicing for spinal muscular atrophy. And then shortly thereafter, the first FDA approved a gene therapy for an eye disease where you just inject the eye itself with a vector, and it can fix a genetic cause of blindness. And then not too long after that, spinal muscular atrophy again in the news with approval of the first systemic gene therapy for for a genetic disorder. And now we've got, as I said, lots of genetic disorders that are being addressed or at least looked at by gene therapy. But no discussion of gene therapy would be complete without talking about Jesse Gelsinger. This is a young man who was diagnosed with a disorder that made him not able to metabolize ammonia in his body, something called OTC deficiency. And it's carried on the X chromosome. Males are typically affected very seriously, and females less so. And Jesse had hyperammonemia, high blood ammonia, that led to his diagnosis as a young child, and spent a lot of time going in and out of hospitals. He was I've forgotten his age now. I think he was 19, but he was the 18th patient that was going to be treated with an adenovirus-based vector, one of those early ones, for his disorder back in 1999. Unfortunately, he was given that vector and was okay for a couple of hours, and then suddenly got horrendously sick and actually died. And this was because he developed an immune reaction to that vector. It's a virus. His body reacted to it, and it went out of control. And that really put gene therapy on hold for many years, understandably, until we as a field could identify vectors that were safer, that didn't cause that. And so, as I said, that was in 1999, and it really took, as you saw, to 2017 or 18 for the next gene therapy for a rare disease to be approved. And all of that time was spent in developing new vectors. And so the two main vectors that you'll hear about these days, something called adeno-associated virus, or AAV, and lentiviral. Either of those can either be used to, for example, fix blood cells and give them back to the body, the way it was originally tried with adenosine deaminase, or inject into the bloodstream or the brain or an organ and try to get expression and fix that problem. The two vectors are fundamentally different in the way they work. AAV does that episome thing that I mentioned earlier. It injects a DNA, a copy of the gene, and then it hangs around and makes the protein that's deficient in an individual. There are immune responses elicited to that vector, but fortunately, for the most part, they have not been as serious as what happened to Jesse. But there is dose-limited toxicity, as we'll see in a minute, that if you give too much of it, you can really develop a problem. The other vector, lentiviral vectors, fall into that category of vectors that are meant to integrate into your normal chromosomes, and so those are the ones that hang around forever. This is a form, the human immunodeficiency virus that causes HIV/AIDS, is a lentivirus. This has no relationship to that, but it's just so you might hear that as an aside. Now, I mentioned that if you're going to make a gene therapy work, you got to hit the target, the organ that's affected. So this can be the brain, and you can and some of the vectors that can be injected through the blood and get to the brain. Others have to be injected directly into the brain or the spinal column. You want to be able to get to several really important tissues. Here I show a liver and heart, But sometimes you have to have other all of the tissues in the body that are repaired, including important organs like skeletal muscle or kidney. So check my time here. I'm going to try to move through this part really quickly because, as I said, it gets a little. Oops, let me bring these guys up. A little complicated. AAV vectors are viruses, or AAV adeno-associated viruses, or viruses that are naturally occurring. They got their name because they tend to hang around with adenoviruses, which you might recognize as a cold virus in the lungs. And so we all have them naturally, and you have antibodies to some of those. They're made in a whole bunch of different varieties, and not everybody's been exposed to every variety. But part of the problem is that if you've been exposed to the variety that's being used to make a gene therapy vector, then you can't get that therapy. So folks are using different, they're called serotypes of AAV vectors or AAVs to make gene therapy vectors. And this is a virus that carries its own genetic information in a single strand. Our information is double-stranded. When it injects information into the cell, it replicates itself. The body makes a double strand of it, and then it hangs around as an episome. Now, why does this work? Well, you can cut out all of the guts of the virus, the genes that make the proteins that the virus uses to make itself, and put in this gene that you want to have expressed. Now, then you have to do certain cellular tricks to make to actually then get active virus out of this. But what ends up happening is you've got this virus that will express your transgene, but not make more copies of itself. And that's the most common form of vector in under study right now. And it looked like that was working pretty well. There were immune responses, but they were manageable until, unfortunately, late last year, there was a virus from a company called Audentes that was being used to treat a disease called myotubular myopathy, and it looked very good in the animal studies. It was working at a lower dose. It's starting to get a response, and they went up to a higher dose. And 3 patients dosed all had very, very severe liver disease, and 2 of those patients died. It now looks like it was probably due to unrecognized symptoms of the disease. It wasn't clear. This is a muscle disease, and it wasn't clear that there were liver problems in it until this happened, and then people started studying it a little bit, and that seems to be what was going on. So the FDA didn't put a stop on all gene therapies, but they did put a stop on this one, and we have to keep this in mind as we're moving forward. I'm going to show you just one example of what this looks like. There's a disease called Fabry disease, which is one of the lysosomal storage diseases. It's a molecule. It's an enzyme that breaks down complicated molecules to recycle them in the cell. And one of those lysosomal storage diseases was the first enzyme replacement therapy that I talked about before. An enzyme replacement therapy for this one wasn't too far behind, and then second generation ones that were better came along, but it didn't address all of the problems of the disease. This is actually a really great model of all of the things that I talked about earlier: chaperone therapy and substrate modulation, in this case reduction. There's an mRNA therapy, but we're going to talk about the gene therapy side of things. We work with a company called 4D, and they have a gene therapy program for Fabry disease. These are data that are publicly available. We presented them at an international meeting earlier in the year. And it's an AAV vector. It's a special one that they've selected to have, they think, broad distribution, because you've got to get enzyme in at least liver, heart, and kidney in this disease. And what you can see here is that Enzyme replacement therapy, if you look at the heart of a mouse with this disease, it gets into cells a little bit. This is this dark color here, but most of the heart cells don't get it. Whereas when they use the gene therapy, in those animals, you can get, now you can see the dark is in lots of different cells. So this can improve the heart disease that happens in these patients. Here you can see that the heart again with the dark being the Fabry gene, and here the kidney. So this looks like a good drug for this disease. Here you can see the heart in a primate model. The FDA wants to see safety data in non-human primates before they go into people. And so now this is in clinical trials. And what you can see here is for the two of the first three patients that were treated. This is the normal level down here, and patients treated with this drug have developed huge levels of enzyme in their bloodstream. It is dropping a little bit, but it's still quite a bit higher than normal. And this patient is the only one that has out long enough, a year, to have some indication of improved heart function, and it does seem, in fact, that he does. So this is a treatment that's ongoing, and a clinical trial being done here at Children's and three other centers participating in that. I'm going to skip this for time. And then just to show you that the lentiviral vectors, even though these are a different virus and they're designed to integrate into the DNA in the cells, aren't really all that much different from what we said before. You have this virus that has its own DNA. You cut out the part of the DNA that makes the viral proteins, stick your gene of interest in there, and then you can have this virus you can have this DNA packaged into viral particles by mixing it in with a viral DNA that will make the proteins necessary, but not package itself. So then you only get the virus that has the right DNA in, and then you put it into cells. I should mention that with that other technology, the AAV, there are ways now that tricks that can be used to try to make that integrate. It's just potentially at a lower efficiency, but there is a clinical trial that's going on where with the AAVs that's also integrating. So that's really quickly the basics of gene therapy. I don't want to go much more into that because I'll spend the last 15 minutes or so doing is talking a little bit about how we bring our understanding of a disease and a potential therapy into a patient. So taking that knowledge and translating it into a treatment. I outlined this in a slightly different fashion in an earlier slide, but you can see the same thing. There's this preclinical phase that I mentioned, but what that is those are the laboratory studies that says this might work. You test it in a cell, you test it in a mouse, or another model, another animal model, and it looks pretty good. And that's what you take to the FDA and say, all right, I want to give this a try. And then you go through clinical trials, and clinical trials have typically 3, and if you call the post-approval phase a phase, that's the 4th phase. So phase 1 is very, very small numbers of patients, and it's really designed to test safety. You look a little bit at efficacy signals, but it's really designed to test safety. Phase 2, now you go into a few more patients, and you do dose escalation, and you start looking for efficacy, and then finally you say, okay, we got the right dose, we haven't seen any safety problems, it's starting to look like it's effective, we're going to go into a larger study, and that's called phase 3, and then when that's done, we go for approval. And then maybe you have this post-marketing surveillance beyond that. And you can see the timeline here. And that's just why it takes a long time and it's very expensive to develop a drug. Now, in rare diseases, we're looking for ways to try to shortcut that process. Preclinical is often done by people like me, people who work in an academic institution, and we're just looking at things based on what we know about the disease, and we can bring a lot of information to bear that doesn't have to actually be done by the pharmaceutical company, and so it saves a lot of time in that regard. The other thing that saves a lot of time potentially is sort of condensing the phases of the clinical trial into overlapping phases. And so we often have what's called a combined phase 1, phase 2, and then a combined phase 2, phase 3, where in the early part you're looking first at efficacy, or I'm sorry, safety, and then you start looking at the efficacy signals, and then you expand your population, but without having to go back to the FDA for a formal approval to phase 3. Now you go to a larger number of patients. But each of those phases tends to be a relatively small number of patients. Most of the time, maybe 2 or 3 dozen patients, and in some cases as few as a half a dozen to get approval. But this really then relies a lot on this post-marketing surveillance phase to better understand what the drug is doing. And even though we thought it was working at the end of phase 3, we see that it's not really changing the clinical outcome. A drug can be, after approval, a drug can be pulled from the market as a result of that. And getting this process in place really requires a lot of advanced discussion with regulatory agencies such as the FDA, so that the protocols are being developed and reviewed right along with the FDA, so that when the time comes for approval, the FDA has already had a say in what the protocol looks like. A lot of the protocols for gene therapy for rare diseases rely on something other than a clinical outcome, because the clinical outcomes are either too short or take too long. They happen too quickly or they take a long time, and they're not good. It's not very easy to get an impact in diseases in a short period of time in that setting, and so we move to the biomarkers that I mentioned, which can be used under certain circumstances, defined by the FDA as what we call surrogate endpoints, things that predict clinical outcome based on what we know about the disease, but can be followed over a shorter period of time and lead to a more rapid approval. And then we get to that post-marketing phase where we now look to confirm that what we thought was happening on the basis of the input, the surrogate marker, is really translating into clinical benefit. Scaling this process is going to be a challenge, and I'll talk to you a little bit about that, but we won't spend too much of time. We have to be able to move from proof of concept to larger trials to approval and distribution in ways that really just aren't present right now. If you think about there being 7,000 different disorders, if we tackle them one at a time, it's going to take a long time before we get to the ball. But what if we could just say develop one basic vector, one basic gene therapy vector, and just plug in different genes? That's called a platforms process, and maybe then you can do 100 diseases instead of one. We need to know a lot about the diseases, right? I talked about the need for clinical data on natural history. Well, perhaps groups that are all interested in the same disease can share their clinical data. And then once they're approved, you got to have the ability to get these out to patients, and we'll talk about that a little bit more, because it's not simple. It's not just as easy as saying, okay, you have a disease, I'm going to give you the vector and off you go. You have to be able to make the right diagnosis. You have to be sure that that the vector that you're using will work with that with your specific version of that disease, your mutation. You have to be able to foresee and treat complications around delivery of the gene therapy, and then ultimately provide the long-term follow-up to show that it in fact is working. And this is an iterative process. So many of us are thinking about this in a way that gets beyond the specialized academic medical center. These are centers that do this on a research basis and are able then to translate it very quickly into clinical care. But it means you have to come here. And so one of the benefits in medicine to the pandemic is we've developed a lot more expertise in virtual medicine, and so that at least helps us at least maybe for some of the follow-up. But even that's not necessarily the best way to go, and so there's a lot of discussion about having hub and spoke models where you have a coordinating center that functions to cover a region, and then the hub, and then spokes that work through that hub. And some of the therapy can be done at the hub, but some of it can be done at the spokes. Or if you look at the Venn diagram on the left, you can see that some centers might be a follow-up center, some might be a treatment center, some might be a diagnostic center, but then there'll be coordinating centers to work through them all. Don't know what that's going to end up looking like, but these are things that we're actively exploring at the Center for Rare Disease Therapy, and in fact are looking now to develop a Center for Gene Therapy as part of the Center for Rare Disease Therapy that will allow us to be able to bring these therapies to our patients and those who surround us, who might not be able to get access to them otherwise. So we're still early on this. We're in the planning and proposal stage, talking to our various components here, including our financial folks, our health insurance plan, our pharmacy in terms of how to safely handle these drugs, and the reality is these are all expensive activities, and so are involving our foundation to help us fundraise around them. We're just getting now into the operations and process component of this timeline, where we're looking to see what the staffing is necessary, how we get this information out to patients, where it's going to live, and then ultimately I think this timeline is really meant to indicate about 3 to 5 years, where we'll be able to expand the number of patients that we see as more and more of these come online. The problem is cost. Here's a graph that shows the cost of not gene therapy, but total prescription drugs in the United States since dating back to 1978. And this is listed in billions of dollars. So you can see this was put together in 2017, I think. And that number was already greater than $300 billion a year. And it's projected to go up to almost $700 billion by 2026. And that's without things like gene therapy, which at least at the moment, the ones that are on the market have all been in sort of the $1 to $2 million a dose range. So that's not sustainable with our current model of medical insurance, and so we've got to address that as we move forward in looking into broader implementation of gene therapy. And the other reality of this is that is in our country, not everybody has access to the kinds of healthcare insurance that's going to be necessary to allow utilization of drugs like this. And so if we're going to be able to deliver these drugs in an equitable with an equitable process and equally across our population, we're going to have to create innovative approaches to providing those therapies, engage our communities of all sorts in helping us to develop and implement those strategies, and in essence democratize access to high quality not just treatment but the educational materials to help people understand what their treatment is going to be like, and so really thinking about building trust, not just in science but in the implementation of science, i.e. medical care. So where do we go next? Well, right now it looks simple. There's only one way to go, forward. But we're getting to a point where it becomes a very, very complicated process, and hopefully we're going to be able to provide you with multiple options to treat rare diseases, one of which will be gene therapy, and then we'll be able to hopefully in a few years come back to you with understanding not only what that on-ramp to this cloverleaf looks like, but what your path forward is from there. So with that, I'm going to stop and say thank you to you for taking the time to listen to this this afternoon. It looks to me like we have oh geez, not very much time at all. Couple of minutes maybe, and I don't know if I can monitor my own chat here, but if you want to type a question in, I'll see if I can see it. I can try to answer it, but if not, I will look forward to you being a part of this process if you're local to Pittsburgh, or you come here for one of those therapies. So let's see. I am not seeing anything coming up in the chat. That may mean that it just doesn't work. I see questions also, so I don't know what the difference between questions and chats are. So I guess we'll just go ahead and conclude. Once again, thanks for being online. If you know friends who might like to listen to this but weren't here today, it will be, it's been recorded and it will be online in, I don't know, probably a couple, 3 weeks at the Center for Rare Disease Therapy website here at Children's Hospital. So you can just Google that and they should be able to find it. So thanks again, and I look forward to seeing you sometime in the future. Bye-bye.